Good evening and welcome to EASL Studio, your weekly hepatology broadcast news. My name is David Jones from Newcastle in the UK. And with me, I have three good friends and colleagues, Bettina Hansen from Rotterdam and Marco Carbone and Lleo from Milan. And what we're going to be talking about today is evidence, trials and primary biliary cholangitis, and try and explain the background to some of the work that's going on at the moment about how we can better show the effectiveness of new treatments that emerge. And this is a hugely important issue in PBC at the moment, but it's relevant, I think, to anybody interested in developing new therapies in hepatology, because this is about the challenge of what evidence do you need and how do you collect it. So Anna, do you want to maybe just introduce a little bit about the consensus process, what it was, why it matters, and how it came about?
Sure. Thank you, Dave. Well, I would say it was a very spontaneous process. If you remember, we had a very kind of a difficult time over a year ago when obeticholic acid was removed from the market. We were getting all these questions from patients. We had already some data, and we were able to identify those patients that had and could benefit from the drug. It was an important period for us. It was the first time that we had something commercially available for treatment of non-responders in PBC, together with the old fibrates. So talking among colleagues, with patients and patients associations, we thought we really need to address this. We cannot just, let it be or be passive on this process. So we reached out to the scientific societies. We thought the best environment was involving the international scientific societies. We reached out to EASL, reached out to AASLD, and also the Latin American Association. And the important thing of this consensus is that we put together a group of people that range from experts to patient advocates to industry partners and regulatory agencies representatives. So all the stakeholders that actually work together and were facing the problem decided to dedicate a lot of time, I would say. We had weekly meetings addressing the main important questions that were actually at the bottom of this very important problem for us and for our patients. So that's how everything started. After that, and after all the weekly work, we hosted a consensus live in Amsterdam, in EASL. And recently in Washington, during the Lleo meeting, we reported on all the recommendations that will soon see the light in a form of a written paper.
Thank you. And I think it's been a really important exercise. And it is fundamentally, in terms of the trial element, isn't it? It's about the concept of surrogate markers, surrogate endpoints, that are those things that are reasonably likely to predict a clinical outcome in the disease. And that's what much of the debate with the consensus was around. So Marco, can I ask you first, obviously everybody, I suspect, in this call will be familiar with liver biochemistry and with transient elastography, liver stiffness measurement. Would you explain the status of those, do you think, those measures that we use all the time in the clinic and which have been widely used in trials? Would you explain the status of them really in terms of clinical trials and their value as surrogate endpoints? Are they useful? Do they have problems? Can we improve them, do you think?
Yeah, sure, Dave. So in PBC, we really rely on liver biochemistry. And for liver biochemistry, we mean, mainly alkaline phosphatase, bilirubin, but also transaminases, GGT. Particularly, the alkaline phosphatase plays a central role as a surrogate endpoint in clinical trials, and it is normally used as one of the primary endpoints in PBC clinical trials. And this is because we traditional hard outcomes like liver transplant or need for liver transplant, liver-related death, the compensation are impractical to study in a rare, slowly progressive disease like PBC. The improvement in alkaline phosphatase and the bilirubin are strongly associated with long-term outcome. This has been largely shown over the last decade in large-scale retrospective studies. And there are scores that we use them in clinical practice and in trials that incorporate this parameter, and they help us to stratify patient prognosis. So as a result of this data of liver biochemistry as predictors of outcome, the regulatory agencies increasingly accept the biochemical response as a surrogate for disease modification, while recognizing the need to further validate these biomarkers so that they be more robustly predict the hard clinical endpoints. And actually ALP and bilirubin are used as a primary endpoint and for drugs, compounds that have received a conditional approval for in PBC. Then we have the transient elastography as well. This is another biomarker, is more commonly used as a secondary endpoint. This adds complementary information by providing non-invasive measure of liver stiffness, and which might reflect fibrosis, inflammation, cholestasis, portal hypertension. And the changes in liver stiffness over time correlate with clinical outcomes. And so they help these help to distinguish patients with a progressive disease from someone with a stable one. That said, they both have limitations. They are distinct indirect markers rather than true clinical endpoints. And improvements of these does not automatically translate into proven survival benefit. And this is the point that the regulators stress a lot. And for this reason, they should be interpreted as instrument, as tools to estimate the risk reduction more than a definitive evidence of disease cure. And so going forward, they're probably integrating the liver biochemistry, elastography, real-world outcome data will be essential to refine how we assess the therapeutic benefit in our patient in PBC.
And I think so. Certainly I look at my own practice and people I talk to, and I'm sure it's true for all of us, is that liver stiffness measurement has become a routine part of what we do. And actually, I think its importance in trials is going to be twofold. It's partly about stratifying people in terms of what treatment they might need. So I think we're all now thinking about, with Christoph Korpeschow and the global group's work on liver stiffness measurement, 10 kilopascals seems to be a very important cutoff. And if people have a value above that, their risk is very high. And so we would think about those in a different way. So baseline stratification, but also, of course, changing liver stiffness measurement as a sort of marker of real benefit. I think it's very interesting here that we've got a parallel with what happened with UDCA in the early days, and I've been around long enough to remember the early days of UDCA, and there were many arguments over many years about whether it worked or not. It seems very strange given what we know now. And actually, it took a long time to show that the benefits in biochemistry you had with it were actually associated with improvements in survival, and one predicted the other. And as you know, because so data, that's now been shown for second-line therapy in PBC as well. I think those are really important observations, and we sort of need to learn the lessons from the past. Just another question, which is that you mentioned the problem the regulators have got, if you like. What is the problem? What are they worried about? We've shown that the drugs improve biochemistry. We've shown that they at least stabilize liver stiffness measurement. And as hepatologists, those are the data we use. What is their concern, and what's the problem, if you like?
So, first of all, as a background, the clinical trials in PBC are typically designed around patients who have failed with ursodeoxycholic acid, who have an inadequate response to UDCA. And so the trials are enriched with patients with abnormal biochemistry, patients with high disease risk. In order to, the aim is to enrich the trial of patients that can reach easily a clinical endpoint test. And this trial design as an add-on trial with an investigational drug on the top of UDCA. Because we said, the liver-related death and transplantation are unpractical as a primary endpoint, the trials rely on the surrogate endpoint. So the main issue with the current way the regulatory pathway is designed is that it demands large and prolonged outcome trials that are not realistically deliverable in PBC, while at the same time placing limited weight on the real-world evidence once the drug is approved. And of course, this create uncertainty around the long-term benefit and has led to regulatory inconsistency, as Anna alluded to the obeticholic acid story. So overall, the system is not well aligned with the biology and the epidemiology of the disease of PBC, and there is a need for a more flexible framework that can integrate, validated surrogates, long-term safety monitoring, high-quality real-world data, and I know Bettina will touch on this.
And I think you're right. This is we all want the same thing. We want the same thing as the regulators of the patients, which is proven safe and effective treatments. It's about how you reach that point. What's the approach in the community about this? How are we as a community dealing with it? How are we coping with this, do you think?
Yeah, well, in the community, there is a broad agreement that relying exclusively on the traditional hard clinical endpoint is unrealistic. Anna mentioned this work that we're doing together with the regulators, industry, and the patient association groups. So there are several proposals that are emerging. First of all, there is lots of work, focused on rigorous validation of the surrogate endpoint. Mm-hmm. So the endpoint we just mentioned, they are not strong enough for a full validation of the new compound, but if we manage to validate and make them robustly associated with long-term outcome, these might validate all the other trials that we've been running over the last year. We need to ensure that the surrogate endpoints, so ALP, bilirubin, have a stronger reproducible relationship with long-term outcome. They need to be we need to show that they are independently associated to the outcome regardless of the drug use, so UDCA, fibrates, new PPAR. And then we need to give a proof that we really understand the mechanism of action. So this, first of all, this could increase the confidence in this surrogate endpoint. Another second kind of work we are doing in parallel is the improving the systematic and prospective collection of real-world evidence from well-characterized patient cohorts, national and international cohorts, according to standard of care, and embedding prospective well-structured real-world data as a core component of drug evaluation, rather than an afterthought, would allow long-term effectiveness and safety to be assessed more reliably and in a representative patient population. And then the third part, probably Bettina will develop this concept, innovative trial design, the use of synthetic placebo cohorts derived from natural history cohort or simulated cohort, using novel technology, the digital twins. These are being explored. And this approach could reduce the need for traditional placebo arms in a rare disease while preserving a robust comparative evaluation of the treatment effect. So together, the strategies might aim to create a more transparent and helpful regulatory pathways that balance the timely patient access with the rigorous evidence of long-term benefit and safety for patients.
And that's led on beautifully. In this exercise, there were three elements to it. The first was to have greater confidence in endpoints in trials, which we've been discussing, in a sense, making traditional trials better. And the second element was, are there different sources of evidence that we can use to either replace trials or to complement them more accurately? And you've very nicely introduced, Marco, the concept of real-world evidence. So Bettina, how can real-world evidence help with this, help solve this puzzle that we've got? And perhaps before you answer that, just for those people who are new to the term, what do we mean by real-world evidence?
Thank you, Dave. Indeed, a very important question. Yeah, I think it's important to start with real-world data. What is real-world data? Because the real-world evidence is the information and the real evidence that we draw from real-world data. And real-world data is not the case that you see in your clinic. No, it's a pool of data where we really try to learn hard what is the outcome, what is the surrogate endpoints that we are watching and should look for, but also what is the association with the hard endpoints. So that's the real world evidence drawn from real world data, from a big pool of data. And how can that help us? I think it's important, and that I think is the most important, is that we actually nowadays with the most modern tools that we have, can actually provide a new framework to support drug approval in PBC, and also to satisfy what comes after this, namely the post-approval, that you can really use the real-world evidence to support post-approval study that we need to do. And I think that that's the way forward in rare diseases, especially for PBC, where the outcome is very far away. It's a chronic disease, and it has all those components that you where you need to think differently and be a little more modern. And the tools are there nowadays to do this.
So, and I think the key thing is going to be the data on which you do the analysis, isn't it? So some thoughts on what the data sources should be for these real world data studies. And this, I think, is important for people in other diseases. This is something we've been thinking about for a while in PBC, but I'm sure in other particularly rarer liver diseases where we may go down the same path, I think it's quite important not to build in errors to the data at the beginning. So now is the time to be aware and think of this. So where should the data come from?
Yeah, indeed. Yeah. Several sources. There are several sources, and one thing that I know the four of us have all been working on is to get the data from sites. So making a registry capturing all patients with PBC in the different sites that we know are having transplant centers, but also the smaller centers where patients are treated. It's so important to have the whole aspect of the disease captured. But there are other ways of having real world data. We have the, what you call, from insurance company claims data, where we can actually go in and see what is the use of treatments and how is the outcome in that sense. And of course, you know that there are different ways of capturing data. It depends, of course, on the hospital, how they are set up, where if you are able to draw that electronically. And sometimes you need to dig in more specific to see what is the treatment that is used, etc. And then you can do that retrospectively, or you can do it prospectively, which have the convenience that with prospective data, you can also capture safety.
And I think that's a really important point in that, particularly where you're looking at comparisons between people who did and didn't have a therapy at some point. Recording the reasons why that decision was made in clinical practice is probably the single most important thing that I've come across, because if there's a confounding sort of reason why they didn't get the treatment, then that will be an issue. I guess that leads on to something I hear all the time, which is the worry that the regulators have around biases in real world data. Do you want to just talk about what the potential limitations of this approach are and how we can identify and minimize those?
I think one thing that's for sure is very important is that if you need to do such make a placebo arm with using the real world data, is that you have the regulators on board already from the start. I think that's really a recommendations that we came up with all together anonymously, all saying that this is important. They need to think along on how we are going to capture the right patients to avoid any kind of selection bias or confounding information bias and all the other kind of biases that you can think of. And then there are processes how to mitigate these things during the whole process of comparison with new drugs. And of course, you need strictly inclusion-exclusion criteria, same area where you look at the patients so that the background population looks the same. So there are a lot of mitigation things that you can do to come around and avoid these and study these biases. So it's not something that is not this is really all possible, and I think that's important.
And I think, and all credit to my colleagues in the international societies who've really driven this forward, I think it's so important that there is a sort of consensus around it and the professional societies supporting it, because getting people to understand the importance of consistency of what you do and having sort of an approach around it, and then taking that learning into other diseases, rather than the sort of historical piecemeal approach where individuals derived these data, having central standards that people agree on, I think is a really major way forward. And as I say, the kind of plea to anybody embarking on this journey in other diseases is learn the lessons from people who've been doing this for a while about what they wish they had done 10 years ago when they set these things up. And it can shortcut this hugely. So I think that's a really important element. So building real world evidence and real world data into the evaluation alongside conventional biomarker driven clinical trials. And the third element of this is kind of very close to my heart, which is around symptoms. So yeah, symptoms in PBC and quality of life.
Yeah, that was the third aspect we really digged into. We know very well that patients want to have a good quality of life. And although for many, many years we've looked at alkaline phosphatase or FibroScan, we realized that it was very important to focus on how we collect quality data on quality of life. So how do you think, Dave, you've dedicated your life to this, how do you think quality of life should be assessed in PBC?
So I think it's really important. So I think, can I just say, I think it is so important that this was included in this exercise for a variety of reasons that will come out. It's also very important for the patients and very positive. And I do think this is an area that will be very productive. So I think in terms of how do you assess quality of life, I think there are two elements. So the first is what do you do in ordinary clinical practice, and then the second is what you do in clinical trials. That's really important. Whereas liver stiffness measurement and biochemistry would be what you'd include in a trial and would also be what you would do in routine clinical practice. That is not true for symptoms and quality of life, because you need to have quite detailed symptom capture in trials that you don't need to do in routine practice. So what I say for practice is just acknowledge the importance of it to individual patients, discuss it, as briefly or as, extensively as you need to in the consultation, then don't get too bogged down. And the one thing I don't suggest people do is use complex quality of life measures in the clinic. There is no need to do that. They're research tools. They are things that you're using in research clinical trials, so don't get bogged down. We just use simple questions. Do you have any itch? Yeah, yes or no. And if they have itch, we ask them on a scale of 0 to 10, where is that? Do they get itch at night? Do they want itch treatment? That's it. That covers itch and gives you pretty much 90% of the information. One of the kind of complex measures would do. Fatigue's a little bit more complex because you have peripheral and central fatigue, but it's all question driven. Most important thing is not to avoid talking about it because you don't think you've got time. It's to introduce it and give people the opportunity to. So that's clinical practice. None of that information would be remotely good enough for a regulator. So you do have to have more detailed information. And so I think this is one of the really powerful parts of this exercise, is to get an agreement on how we might structure what we would call a package. So you use a number of measures to assess different aspects of quality of life, but to get agreement about the ones we're going to use and to standardize their use so that we aren't pulling together different, we all use the same alkaline phosphatase, but in symptoms you can have lots of different measures. And I think where this exercise is very useful, the consensus is that you have a PBC specific measure. You then, be that for itch or fatigue, you then have below that a cross-cutting symptom measure that will allow you to compare severity of that symptom with other diseases. And then below that, you have what's called a health utility measure, which is a measure of functional status. And that's really important because that's what the reimbursement people want to see, so you can understand the health economic value. And so a standardized approach, a PBC specific measure, a generic symptom measure, and then a standardized health utility. And the recommendation is that we use the standard package in all trials moving forward. And I think that's a really important step, and it will make life easier for people, because unless you're an aficionado of these measures, it can become a word soup very, very quickly.
We are also facing a time in which the drugs we have available are able to improve quality of life. Yes. And on the other side, we also have some drugs that are being developed targeting as a primary endpoint quality of life itself.
Yes. Yeah.
So do you think the assessment of quality of life itself will help the regulatory pathway in PBC? Will we see a change in paradigm here? How do you see the future?
And I think you've alluded to what I think is a major opportunity. So if the FDA was saying, what's the grounds for approving a drug, they would say it's got to improve survival, how people feel or how people function, feel, function or survive. And of course, we're all used to survive, aren't we? We want our people to live longer. But in a disease like PBC, it's very difficult because that's in the future. But how you feel and function is tomorrow and the day after. So you take away the lag of the impact of a therapy because you will see a direct benefit. So it shortens it all. So therefore, it becomes very important because you can see quantifiable benefit. The challenge is that even I wouldn't accept that symptomatic aspects are as important as survival. So this isn't seen as important, and I would agree with that. And so you are arguing for a value that is different to improving survival in people, but it may well be one that is easier to measure. The one thing I think that people need to do is to be clear where the benefit lies with the therapy. And I think we have seen a little bit of therapies being sort of secondarily evaluated when used for kind of improving survival to look at their quality of life benefit. And I actually think some of the symptom benefits are bigger than that, and they warrant being looked at in their own way. So actually, I think this may well be a route to regulatory approval of drugs. And I'm fully aware that there are colleagues in the PSC world who are using some of the PPARs, bezafibrate as well, as drugs to treat pruritus in PSC, but they are really using it because they believe that they will improve survival in the disease. So we can get access to treatments on this basis. So I think it gives us another string to our bow. So I think it's an opportunity, and I think industry get this. The other thing I would say is looking at symptoms quality of life is very important. Patients sort of value this, as a way to do things. So thank you for including it. I think it's very important. I guess my last question was going to be for all of you, which is crystal ball time. In 3 years' time, when this has all worked its way through and a new therapy comes along in PBC, what's your best guess as to what the evaluation would look like? You've done early phase trials, it clearly improves biochemistry, people feel great on it. Will we be doing a traditional trial, then trying to do confirmation? Will we be doing a blended trial with synthetic controls? I guess it's the where does this kind of end up as a kind of destination? Anna, I'd be interested in you've been steeped in this from the very beginning. Do you think we're close to a breakthrough, or do we think we're in the discussions about a breakthrough?
I think we are still in the discussions, right. We are getting close. At least we are having these discussions all together, involving all the stakeholders. And I think that's been key in all this process. We need to move a step forward, and we need to start working together in a design that in 3 years will lead to something completely different.
Marco?
Well, I mean, the low-hanging fruit will be validating the ALP and bilirubin as a surrogate endpoint, and so we'll use them as a primary endpoint for fully approved drugs. The difficult part will be to have to be able to validate, the real world evidence, and or to create this blended platform with simulating real data. That I find it very challenging, and I think we need lots of, we need to help each other with the scientific community and the regulators. So this community work is really important.
And Bettina, is this the beginning? Is the future yours with the real world evidence models and the blended models, do you think?
It's Christmas, huh? So I can make a wish, right? So if I get my present, I don't know, but I can wish for it, and I definitely, I think that there is actually, yeah, lights ahead of both what Marco is referring to, surrogate endpoints approval, and also using real world data. We are far with this methodology. It's solid. It's really solid. So yes, I do believe that we'll get there. And I think the most important is that we collaborate all together to get there.
And I would say that obviously the UK is no longer regulated by the European Medicines Agency after a little sort of local difficulty that you're all familiar with. However, the MHRA, who is now our regulator, have just put out a discussion paper about incorporating real-world evidence into trials. So the UK is about to embark on this journey, informed by all of the activity that we've been doing and other people. That's not about PBC, that's about rare disease. So I do think there are people who are prepared to be looking at all of this. And I think you deserve particular thanks from the community because of the work that you've done. You're very modest about it all, but you were the people that showed that URSO really worked. And believe me, I lived through those arguments and I don't particularly want to do that anymore. So I share your optimism. I think the future looks very bright. We hope you enjoyed this. The reason for doing this is twofold, because the document is coming out shortly. And this is just a little bit of background as to why it happened, why I think it's important. I think it's really important, and what we did and why we did it. And the fact that getting the consensus around it has been so important, and it does present a framework. And as I said at the beginning, for those people who are not sort of steeped in PBC therapeutics, this has important lessons for people in other diseases where you might be embarking on these journeys. Get the real world evidence structure right at the beginning and it will pay dividends down the line, and learn how to blend it with conventional trials. And don't forget the importance of symptoms as an important aspect for patients, but also as a sort of regulatory route forward. And with that, our time is up. Thank you very much for joining us. This is the final episode of the season. Thank you so much for tuning in, and we all wish you a very happy holiday. Thank you very much.
Happy holiday, everyone.
Happy holidays.